Yeah, because all the in-person was on Seattle, all the people were inside the game. Yeah. It didn't even open up. It can't be that electric even, you know, it'd be cool, but back. What is it? Distance. Yeah. Hard to yeah. I agree. Oh man, that was exciting. It cuts our offense still pretty thin. Buffalo Bird play, we're on 1 yard, Buffalo away. Gosh, then a couple of handoffs are going everywhere. You turn one way or the other. All season.
We have a policy now whenever anybody walks double time and gets their I was up there one day a few years ago, and it's like the morning.
But then you don't go to the
we do have 4 ambulances in a row coming off the one day set. We're never going to right after he didn't get it that great play. I was successful getting both many.
Yeah, okay.
So you got the information.
Thank you very much.
You're welcome. You know who else loves in the press? Oh, it's not one of these just
Field reimbursement manager.
Actually, she can.
Yeah, let's get started. Glenn's out today. He's watching remotely, so I'm going to yell at me if I don't spend some time.
If you don't mind.
So we went down to Portland.
The word of the day today is colonization.
So
there may be a Seahawks themed one coming, so next week maybe. So Paul was nice enough to get up at 4 in the morning to make it down here to talk to us and talk to us about the microbiome. So.
Thanks, Drew. Good morning, everyone. Coming in on this day after the holiday. It's like a Monday. I was telling Drew that this is an interesting topic, and I hope to just probably cover the surface of it. In the last 2 years, there have probably been about 70 articles published just on this topic alone, immunology and allergy and the microbiome. So any segment of this could make an entire journal club, so it'll be really interesting. Disclosures, I really don't have any. The only therapies we might talk about are probiotic supplementations, and I'm not sure if those even require FDA approval at this point since they're supplements. Just cover some definitions really quickly. These are listed on your handout. But when we talk about the microbiome, we're really talking about the corresponding collection of bacterial genes inhabiting the human host. Some people actually include the human genome in the microbiome definition. Microbiota are the collective bacteria either in the gut or the skin or the respiratory tract. Prebiotics are non-digestible carbohydrates that selectively stimulate the proliferation of specific genera of beneficial bacteria in the gut. Probiotics are live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. It's a WHO definition. Symbiotics are mixtures of pre and probiotics, and dysbiosis is imbalance in the microbial composition and a shift in their function from normal to disease. Just a little trivia to start with. The total number of microbes living in the GI tract is about 100 trillion, which is about 10 times the number of human cells, or anywhere from 800 to 1,000, or perhaps even slightly more species. Estimated cell numbers in the GI tract, 10 to the 12th per gram of contents, 100 times more genes than the human genome, 5,000 different species in the respiratory microbiome, and a set of 28 different species. The major phyla in all of the microbiomes are the Actinobacter, which include bifidobacteria, which is an important bacterial strain in the infant, Firmicutes, which include the Lactobacillus and Clostridia. Proteobacteria contains a variety of pathogens, including E. coli and Bacteroidetes, obviously Bacteroides. With Firmicutes and the Bacteroidetes are the two predominant phyla. Out of 55 different phyla of bacteria, it's amazing to think that there's really only 2 that sort of dominate in the human microbiome. And the individual microbiome really only consists of about 15% of the 1,000 species that are available. And so we'll talk about it, but there's a lot of inter-individual variability within the microbiome. And in a sense, as we'll mention, it's almost like your fingerprint. What are the functions of the microbiome? Well, they have metabolic functions. They produce, they help digest certain non-digestible carbohydrates, resulting in a variety of fatty acids, including short-chain fatty acids, which have significant immunomodulatory effects. And they also are sources of a lot of vitamins. They metabolize proteins and bile acids and harvest nutrients in the gut for our benefit. They also have a structural role. They are involved intimately in epithelial cell growth and differentiation, and they also help degrade mucus and mucus proteins. And they have a protective effect. They induce maturation of host immunity, protect against pathogen invasion, and help maintain epithelial barriers. Some of the older studies, probably prior to mid-2000s, used bacterial culture techniques. And it turns out that only about 1% of the bacteria in the GI tract are identifiable by culture techniques for a variety of reasons. Some of the early culture-independent techniques included FISH and DGGE, and current techniques are using measuring the 16S ribosomal RNA using high-throughput sequencing. 16S ribosomal RNA is unique to bacteria. It's not present in eukaryotes, so when you measure it, you're really only measuring bacterial or fungal or yeast RNA, and it has a variable and conserved portion with the variables specific for taxonomic identification and the conserved portion they use to bind primers. So that helps them, but it only analyzes down to the species level, and it doesn't necessarily differentiate between living and dead organisms. The future probably is metatranscriptomics, uses RNA, other RNAs rather than DNA, which is already replicated, and will be able to get you down into some information at the strain level. But there's really a lot of medical informatic blockage, and because of the huge amount of DNA that you're dealing with, and the amount of culture necessary to analyze those. So when we're going to talk about the microbiota, we're really going to talk about the barrier microbiome, so the respiratory, intestinal, vaginal, and cutaneous. But there's also the environmental microbiota, which has an impact particularly on the cutaneous microbiome. And the environmental exposure reflects on the diversity, dose, and timing of colonization of the other microbiomes. There are a lot of variables involved in an individual microbiome. Age, it changes, and we'll cover this from infancy into adulthood. Body site, different portions of the body have their own little ecological niches for bacterial communities. Method of birth has a major impact early in infancy. Diet can have a major impact. Use of antibiotics prenatally or in early infancy. Mother's immune status, mother's stress levels, the environment, ethnicity, geographical location, microbe interactions, and microbe-host interactions all play a role in the composition of the microbiome. And the important thing about the microbiome is its ability to maintain colonization resistance or competitive exclusion. That's what kind of acts as a protective factor and keeps the pathogens from taking over and causing disease. The other thing about the microbiome is it's unique to individuals. It's equivalent to your fingerprint, but some people feel that there's a shared core microbiota and microbiome, so that everybody has a certain conserved sort of core microbiome between them, and that may be required for the correct functioning of the genome. So this is kind of the development of barrier microbiomes. Maternal factors, as I mentioned, are important with mother's intestinal flora, her health status. For example, allergies. It's the microbiome of a mother with allergies is different from the microbiome of a mother who doesn't have allergies. And since mother's the major responsibility for the microbiome of the neonate, you can see how allergies may transmit through generations to a large extent. Mode of delivery has an effect. and we'll cover that a little bit more. Breastfeeding, and then the maternal microbial communities that are involved in the development of the microbiome are her skin, gut, and vaginal communities. So at birth, it used to be thought that basically the intestine was sterile, but with these new methodologies, it's found that there really are some microbial contents in a newborn's GI tract even before delivery and birth, and the amniotic fluid may not be sterile as it was originally thought. And then it rapidly increases until they reach close to an adult microbiome about 3 years of age. And you start to see an increase in diversity over this period of time, but there are ethnic differences. Westernized societies have less diversity, which may be part of the problem for the increase in allergy and allergic disease in Western societies. There's a tremendous amount of inter-individual heterogeneity, and diet, health, and antibiotics play a significant role in changing that. And then when you get to be an adult, it's a fairly stable environment within your body, but of course you continue to have an inter-individual variability. So in utero events or exposures can actually modify gene expression through epigenetic mechanisms. Initially, the infant GI tract is populated by facultative anaerobes, and these are examples. But by 1 week, the consumption of oxygen by these facultative anaerobes results in a shift to more strict anaerobes with bifidobacteria and lactobacillus to a large extent dominating, but Bacteroides and Clostridium are there. And then finally, after weaning, when the infant starts having solid foods and cow's milk and formula, you start to see a shift into the adult-type microbiota. And the baby achieves that by about 1 to 3 years of age, and it kind of depends on feeding and method of birth. It determines what age they actually achieve that. These are the different factors that can be involved in colonization. For vaginal deliveries, it's maternal gut and vaginal microbiome that are playing a role. In cesarean section deliveries, it's parental skin and the non-living environment, food and other, for example, nursery staff that may be involved. And so you see with C-section deliveries that there's much more Clostridia, Klebsiella, and Enterobacter because it comes from the non-living environment and other people in their environment, and the skin causing staph. Whereas when they're born vaginally, maternal gut and vaginal microbiomes are responsible for establishing the infant microbiome. And the diet has an effect too. This is a table that basically shows some of the differences between breastfed and formula-fed infants. And you can see that in breastfed infants, there are more staph and lactobacillus. In formula-fed, they have more Clostridia, Enterobacteriaceae, and so on. So the other thing about breastfeeding is it supplies prebiotics. So there are oligosaccharides in breast milk that serve as prebiotics. Fatty acid intake can play a role in shaping the population dynamics of the microbiota. The Western diet is felt to have a significant impact on the differences between our microbiome and those in underdeveloped countries. And changes can occur fairly quickly. And what happens with the diet is you can alter one or more species, and then you get alteration of the competitive exclusion properties of the microbiome. And the protective effect of breastfeeding may not apply if mother is allergic because her microbiome is different. Therefore, the baby's microbiome becomes more of an allergic person's microbiome early on. And that may explain why you see this differences in these studies on the effect of breastfeeding and allergy, maybe because they don't control very well for that. And there's a family history of allergy that changes the outcome of a lot of these studies. So what do antibiotics do? Well, they kill large populations of microbes, but probably more importantly, there's a decline in microbial diversity and a decrease in colonization resistance, which allows either endogenous or exogenous pathogens to take over. Changes can last for months in infants. They don't last as long as in adults. and can also occur if mother is treated with antibiotics prenatally. Again, mother's the source of the baby's microbiome, so you change her microbiome, you change the baby's microbiome. Some of the changes in colonization due to Western lifestyle, you see less diversity and complexity. There's less transfer of bacteria from the maternal gut with a result of delayed colonization, increased opportunists from the non-living environment and bred skin due to the high rate of C-sections, deliveries, decreased intake of dietary fiber and vitamin D has a definite impact on the microbiome, and a loss of some of the evolutionary bacteria, and we'll talk about the importance of that to come. And you can see some of the bacterial changes. This 8 categories evolutionary bacteria that can have a profound effect on the development of the immune system. So you just see less of that and less of that with subsequent generations. So diversity is very important to the intestinal ecosystem, and it also relates to spatial topography. So different parts of the GI tract and different parts of the skin and respiratory tract all have different colonization depending on what portion of the tract you're talking about. So that's what we call the spatial topography. There are shifts in species and strain composition over time related to environmental exposures and antibiotics and changes in diet. So there's a high degree of functional redundancy is required. Metabolic function really depends on individual species, but also cooperation between the species in the gut. So the presence of 2 species can be synergistic in some of those metabolic effects. Genes may be more relevant than the species themselves, and the development of the immune system appears to depend on a balanced, diverse microbiota. So just to talk a little bit about the intestinal immune system, and this could be, of course, a topic, I think, a very interesting topic for a journal club, particularly as it relates to the microbiome. But it considered the gatekeeper of the immune system. Main functions are the epithelial barrier and development of tolerance. Components are mucus layers, epithelial barrier, specialized epithelial cells, antimicrobial peptides, and secretory IgA. Should be SCP, rather SIgA. And then the cellular components are dendritic cells, macrophages, T cells, Treg cells, and important parts of the innate immune system, the TLRs and NLRs that are involved in some of these responses with the microbiome. And this is just a pictorial of that. The epithelial layer, IEC, are intestinal epithelial cells. The lamina propria contains dendritic cells and macrophages, Tregs and B cells, naive T cells. There are Peyer's patches overlaid by specialized epithelial cells called M cells that can actually transport bacteria into the Peyer's patch. And of course, some of the dendritic cells are specialized, and they can actually extend the dendrite through the tight junctions to sample some of the bacteria in the intestinal tract. And then they all meet in the mesenteric lymph node and have their convention there. So the immune system is really dependent on the microbiota post-immune reactions, and it's actually bidirectional. If you look at animal models and humans with immune deficiencies, they have different microbiomes and changes in their microbiomes. So the absence of immune components changes the microbiota composition, as well as changes in the microbiota has effects on the immune status. And some of those changes in the microbiota can lead to GI inflammation, which they think is a significant problem with metabolic syndromes and obesity. Gut microbiota are crucial for the development of the gut-associated lymphoid tissue. Most of this data comes from germ-free mice, but there's some interesting specific bacterial products that have a direct effect on the immune development. For example, Bacteroides produces a polysaccharide A, which promotes Treg development directly. And germ-free mice have an underdeveloped gut-associated lymphoid tissue. They have less IgA-producing B cells, less T cells, decreased lymphocyte central locations and germinal patches in the Peyer's patch and lymphoid follicles, and less dendritic cells. And the secretory IgA repertoire is shaped under the influence of maternal microbial exposure as well. It's also crucial for the development of tolerance. Germ-free mice, it's really almost impossible to induce tolerance in those animals. They have impaired Treg function, they have lower IL-10 and TGF-beta, which are crucial for the development of tolerance. And the interesting thing is that you can induce tolerance capabilities by simply transfusing or basically changing the colonization of germ-free mice. If you give them a normal mouse microbial colonization, then they restore that ability to develop tolerance. And the important thing to think about, we don't think about it very much, but much of our inhaled allergens end up in the GI tract. So this tolerance applies to airborne allergens as well as food allergens. The GI microbiota are required for normal intestinal epithelial development. They upregulate gene expression for antimicrobial peptides. And germ-free mice have an immature mucosal epithelium. Structurally, they have a thin mucus layer. They don't have a well-developed epithelium. It's not replacing itself or dividing very quickly. And again, if you just transplant a normal microbiome, you reverse a lot of these changes in the germ-free mice. And missing or altered intestinal colonization can also affect immunologic mechanisms at distal sites. So, for example, germ-free mice have increased allergic airway disease. They have a much more robust allergic airway response. If you transplant normal microbiome in neonatal mice, you prevent that increased allergic development. But it doesn't work if you do it in adult mice or mice after their weaning stage. So again, allergic immune response is dependent on the microbiota, which we talked about, can be converted to more normal responses by reconstitution of the microbiota. And then commensal bacteria might actually induce an anti-inflammatory response. So we talked a little bit about short-chain fatty acids. They're ligands for a G protein coupled receptor called GPR43, and deficiency in this receptor is associated with exaggerated allergic airway response in a mouse model. Lectins derived from carbohydrate digestion by the microbes will regulate immune reactivity through both C-type and S-type lectin receptors. And human maternal colonization with normal commensals correlates with IL-10 production from cord blood mononuclear cells. Recognition of commensal bacteria by toll-like receptors and other pathogen response receptors are indispensable for the allergy protective state. TLR2 is a crucial toll-like receptor that is directly involved in control of epithelial barrier function and repair, activation of dendritic cells and other antigen-presenting cells, leading to the induction of Th1 responses and direct activation of FOXP3 positive Treg cells, all through the toll-like receptor 2. TLR4 is also important. Binding of a lipopolysaccharide to TLR4 leads to increased IL-10, IL-12, and interferon gamma, and inhibits allergic responses. Now that's crucial to occurring before allergic sensitization, because after allergic sensitization it actually leads to amplified Th2 responses. And the TLR activation on epithelial cells leads to the secretion of APRIL and TSLP production by the epithelial cells and responsively by the dendritic cells, and that's important for T-cell independent IgA2 class switching. And TLRs on the dendritic cells may also promote anti-inflammatory responses mediated by IL-10 and TGF-beta. And the response may depend on the variety and duration of stimulation. And there's a little bit of issue related to this, because if you think about it, the pathogen recognition receptors have limited repertoire. And all bacteria, pathogenic or commensal, will have the same sort of molecular or microbe-associated molecular patterns. So why is it that the GI tract and the immune tract responds differently to pathogens as opposed to commensal bacteria? And the answer isn't clear, but it really may be a combination of the variety of different exposures from the variety of microorganisms and also the duration of the intense response. So it's a continuous exposure over time.
Have the pediatricians bought into this enough to give these kids like a bug cocktail when they're young if they get
Well, we'll
C-section?
talk about the problems, and I mean, there's a lot of studies looking at bug cocktails, and there's a lot of problems and variable results. So everybody's thinking about it, but nobody's found the right cocktail.
So you're not going to switch your formula to yogurt or anything like that?
Well, you know, for years they've always tried to make the formula as close to breast milk as they can, but again, what's the right combination? It doesn't matter on the individual, ethnicity, environment. You know, what makes a difference? There's still a lot of studies to be done.
And in a way, this is a study of ecology. If you show up in a pond in the wintertime, there's no grass. There's nothing living here, and you come back later, and there's no organisms. But the reductionists would say, let's just give it grass seed, and it'll solve the problem. You know, you don't really realize that something else might not causing that to be the way it is. So
Right.
I'm kind of curious, like with the biphids, you know, just give Bacillus, you're going to solve all the problems, but there's thousands of other bacteria that aren't accounted for.
Well, and part of the problem, and we'll get into that a little bit later, so I don't want to head that off. But tolerance is an interesting topic that, again, could be a journal club on its own. But mucosal dendritic cells are very important because they induce a tolerogenic B and T cell responses. They facilitate IgA secretion. And there's a specific type of dendritic cell found in the lamina propria called the CD103 positive dendritic cell. It's found in the lymph nodes and the lamina propria, and it's capable of converting naive T cells to FOXP3 positive Treg cells. And interestingly, there's a dietary component called retinoic acid that can be involved in this. And some of these responses are dependent on retinoic acid and TGF-beta. This is enhanced by the C lectin receptors, which bind sugar molecules. And if you look at bone marrow-derived dendritic cells and you incubate them with lactobacillus, you enhance the ability to promote Treg differentiation in vitro. And after adoptive transfer, it prevented inflammation. And these are TLR2 and dendritic MyD88 pathway dependent functions. This is just kind of a summary of some of the adaptive immune response in the microbiome. We talked about the innate immune response. These also are dependent on intact TLR signaling pathways, but the emergence of T cell subsets, differentiation of gut B cells are all important. Effects of the microbiota through the adaptive immune system. Certain microbial signals can actually induce mucosal tolerance by inducing the formation of inducible Treg cells, and polysaccharide A from B fragilis appears to have that particular ability. There is a correlation between colonization in early infancy with lactobacillus and bifidobacteria in IgA, IgM, and IL-10 levels in cord blood. And in, not cord blood, but in peripheral blood mononuclear cells of infants. And specific strains have the ability to induce Treg cells and attenuate allergic responses. And this is just kind of a cartoon of some of the things that we've been talking about. Outer mucus layer is where most of the bacteria are. There's select bacteria that can penetrate the inner mucus layer. These are the specialized dendritic cells we talked about with dendrites that extend between the epithelial cells. There are other secretory C-type lectins. The Reg3 gamma is an example of those that bind gram-positive organisms. And, of course, the response of the dendritic cells through esoteric lymph nodes and interaction with B and T cells leads to IgA production. And this is also a response, a cartoon of basically the response of the human to the bacteria, direct effect of microbiota on inducible NKT cells, innate lymphoid cells, Th17 cells, and Treg cells. And we don't have time to go into that, but different bacteria are associated with different effects. For example, the segmented filamentous bacteria can induce Th17 cells in the lamina propria. Clostridia has an effect on increasing Treg cells. So the respiratory microbiome is in a lot of ways very similar to the gut microbiome. You're dealing with an epithelial barrier, you're dealing with a mucosal immune system, and you're dealing with microbiota. There's a dynamic relationship between respiratory host defense and microbes, and a need for discriminatory tolerance. There isn't as much information on the respiratory microbiome. It's a little harder to sample. It's much easier to sample the GI. But there are different commensal organisms in the nasal microbiome, the oropharyngeal microbiome, and they're actually, the lung is not really sterile. If you do these advanced DNA non-culture or culture-independent technique, you find that there are commensal organisms all the way down to the alveoli. The lung is similar to the oropharynx with smaller quantities of bacteria. And as I mentioned, there's some spatial heterogeneity, so the larger airways have a different microbiome than the smaller airways and the alveolar areas. Nasal has a combination of kind of skin and oropharyngeal microbiome, and oropharyngeal is strep, Haemophilus, Neisseria, and staph. But there's also niches in the mouth where you have different bacteria, the teeth, gums, tonsillar crypts, and so forth that will have variability. Same CD103 positive dendritic cells are present in the respiratory lamina propria, and germ-free mice have less of those in the lung as well. There are decreased numbers of plasmacytoid dendritic cells and alveolar macrophages in germ-free mice in the setting of allergic inflammation, so again suggesting that the microbiota has an influence on lung immune responses. And interestingly, surfactant proteins can actually assist in controlling inflammation through innate immune responses. The skin microbiome is a highly diverse commensal communities along distinct topographic skin sites. So dry skin tends to have the highest diversity, the most impact from environmental exposures, and the lowest number of bacteria. There's also frequently transient changes in the dry skin microbiome. The moist skin, primarily staph and bacteria. So flexor creases are considered moist skin. Staph is important in atopic dermatitis. It tends to pollinize moist skin, so maybe why flexor creases are predominant sites for atopic dermatitis. Sebaceous skin also has distinct bacteria. There's over 100 distinct species, about 1 million or 1 billion microbes per square centimeter of skin, and it varies by occupation, and use of cosmetics can also change the skin microbiome. They have immunoregulatory effects just like the GI microbiome, and cutaneous inflammatory disorders have been associated with dysbiosis or alteration of the skin microbiome. There's a lot of inter-individual diversity, just like the GI tract. Some of the studies in mouse models show that the skin microbiome can have an autonomous role in controlling the local inflammatory milieu and tuning resident T cell function, and protective immunity to a cutaneous pathogen is dependent on the skin microbiota. So what about the gut microbiome and atopy? We're finally getting to the topic here.
Can I ask a quick question before you go on?
Yeah.
The organisms you mentioned in each of those sites, obviously a lot of them we think of as pathogens, Bacteroides, Clostridium, Haemophilus. How do we know, or what determines when they're protective and when they're pathogens, or how do we know that they're actually living in the sites that they are, and we're not just seeing DNA and RNA fragments, and maybe that's the protective?
Well, that's the problem. We don't really know whether they're really living there or we're just seeing DNA fragments. It's the limitation of the technique. The advantage to using these new culture-independent techniques is you discover that there's 100% more bacterial species that you didn't know about. The problem is that you can't identify at the strain level, and you can't identify whether they're living or dead. So we haven't defined what a normal microbiome is to a certain extent, and that's part of the problem with developing probiotic cocktails. If you don't know what normal is, what are you aiming for? You don't have a target. So, this needs to be, better computing capability to analyze and handle the amounts of DNA that you're finding.
Along with Matt's comment, I mean, if you can't grow these things out of the tissue in the terminal airways and the alveoli, is it possible that it is just fragments of DNA and that there isn't really any bacterial flora there?
Well, it's possible, except that they're consistent findings and they're over time in individuals and stuff. So, and there's some uniqueness, and the absence of those causes disease. So the studies in germ-free mice suggest that those are important. Commensals. But again, we need to develop better techniques to really know the real answer to that. There's a lot of indirect evidence, but not direct evidence. So the hygiene hypothesis has now become the microbiota hypothesis. And decreased exposure or altered microbial antigens causes immune dysregulation that's observed in Western society. So atopy, obesity, autoimmune disease, inflammatory bowel disease, all of those relate to dysbiosis or abnormalities or alterations in the microbiota. And of course, diet and antibiotic exposure, which is very different in our society compared to underdeveloped countries, also have a significant role. Cesarean section delivery may be responsible for, well, it's definitely responsible for a change in the microbiome. It causes a delay in normal neonatal microbiome by up to a year. Interestingly, if it's an emergent C-section, has different outcomes than if it's a scheduled C-section, possibly related to maternal stress and also hormone exposure during the process of labor and delivery may have an impact on the microbiome. But studies, epidemiologic studies, have suggested there's a 20% increase in asthma in infants who are born by C-section compared to vaginal delivery. There is a critical time window in early life. Most of the mouse experience suggests that it has to occur before weaning. You have to make these changes and try and achieve a normal microbiome prior to weaning for it to be effective. And a key determinant in the development of atopy appears to be the initial composition of the microbiome. Animal studies with notobiotic mouse models, especially the germ-free mice, show an increased allergic airway response with altered microbiota, which can, and again, this is a repeat, can be prevented by establishment of normal microbiota in very young mice or exposure of pregnant germ-free mice to normal microbiota before the delivery of the infant mouse. Germ-free mice cannot develop oral tolerance, but it can be restored by colonization, but only in the neonatal period. And mice lacking TLR4 or CD14, which is another high-affinity lipopolysaccharide receptor, are susceptible to food allergy.
What's gnotobiotic?
Gnotobiotic are mouse models where you can manipulate the GI flora and just, change species, strains, total absence, manipulative mice models. Microbiota differs in food allergy-prone mice, changes with sensitization, and interestingly, it can be transferred via microbiota. So you take a food allergy-sensitized mouse and take their microbiota and transfer them to another mouse model or another mouse type. That mouse type becomes food allergy-prone just by transferring the microbiota. So there's a murine model of cow's milk allergy. Some people say, well, is it the allergic disease that changes the microbiota, or is it the microbiota that changes the allergic response? And so in looking at cow's milk allergy models, they don't find any change in the microbiota just by causing the cow's milk allergy. So that would suggest that it's the microbiota that are playing a role and not necessarily the allergic disease. If you give mouse a Clostridium cocktail, you can increase their Treg percentage and numbers. You can increase or decrease their allergen-induced airway hyperresponsiveness and inflammation. They have less inflammatory cytokines in the lungs. They have more IL-10, more TGF-beta. Rats with ovalbumin sensitization fed a bacterial lysate that contains these particular bacteria. And it's not important, again, to worry about the particular bacteria because it's going to vary in individuals. But by feeding them a bacterial lysate, they have a decreased allergic response accompanied by an increase in IL-10. There's felt to be a gut-lung axis, so you manipulate the microbiota, the GI microbiota of mice with antibiotics, which don't really affect the respiratory microbiome very much. Or if you give them probiotics, you can either increase their susceptibility or decrease their susceptibility to airway allergic disease just by manipulating the GI tract. Sorry, I lost my place here. Okay. And tolerogenic responses from the gut via the gut-associated lymphoid tissue can spread to the airways in response to the challenge. Administration of natural or synthetic TLR agonists trigger an associated protective effect on experimental asthma and related to Th1 responses. Could also be achieved by the intranasal application of typical commensals in adult animals with protection in the offspring. So again, another evidence for transfer of this immune response. The bacterial strains, though, appear to be different in effect. And you do see increased TLR expression in maternal lung, which leads to epigenetic modification of interferon gamma genes, which results in increased interferon gamma in mother as well as the offspring. H. pylori, you can decrease cellular and humoral immune response by feeding them H. pylori due to an increase in FOXP3 Treg cells. Again, the MyD88 dependent pathway downregulates IgE production, and other epigenetic modifications that have been noted include early colonization with normal microbiota. Reduces the methylation of genes encoding chemokine ligands, which are required for inducible NKT cell homing, so that decreases inflammation. So this is just kind of a study moving to humans now. You can see that maternal factors we've talked about, the allergic phenotype, intestinal colonization, mode of delivery, and breastfeeding all play a role at birth in the development. Healthy microbiome has increased diversity. An allergic microbiome has less diversity. Probably talked about the issue of cause-effect relationship, but there are certainly some studies that suggest that it's a cause-effect relationship and not just incidental to allergic disease. And this leads to, of course, allergic conditions. What are the studies in humans? Well, most of them are indirect or epidemiological, and there has to be some caution in interpretation due to variability between individuals and their microbiome. We know that there are several studies that suggest that their allergic individuals have a different microbiome than non-allergic individuals. And the interesting thing is that that is present before the onset of allergic disease. So the microbiome at 1 week and 1 month of age has been associated with atopy at 1 and 2 years of age, or atopic dermatitis at 1 and 2 years of age. So those changes precede the allergic disease. And they actually may overwhelm the environmental influences. So you may see the same changes in different populations in different environments. Normally, you would expect the microbiome to be different. But with the allergic, there's more similarity between different populations. And there's definitely decreased diversity in infancy as a result of disease. Differences, as we said, in colonization vary with the atopic status of the parents. A lot of stuff about animal exposure and farm effects that we'll try and get into a little bit. Maternal exposure to cats and dogs is associated with decreased cord blood IgE, increased number and quantity of cord blood Treg cells, and a decreased incidence of disease. H. pylori seropositivity is associated with a decreased risk of childhood asthma and atopic dermatitis. And there's epidemiologic evidence that modulation of the gut immune response can directly affect the development of allergic diseases at other sites, so the lung and the skin. The farm effect. There's a lot of stuff written about the effect of growing up on a farm and allergic disease and that's been evaluated to a little larger extent by doing some more prospective studies and analyzing the microbiome to a large extent and what those exposures specifically are. So continuous LPS exposure during pregnancy and infancy is associated with lower prevalence of atopy and asthma. Consumption of farm milk and exposure to gram-negative bacteria facilitate that infection, and you can see some of the immunologic changes associated with that. Maternal exposure to environment rich in microbes upregulates the TLR receptors. Maternal exposure during pregnancy to farm animals and cats is protective against the development of allergic disease. And again, you see elevation of gene expression in cord blood of some of the TLR receptors. The levels of TLR2 and 4 at school age correlate with the number of farm animal species the mother had contact with during pregnancy. But the protective exposures may be different for different allergic diseases. So some of the better analysis of these studies suggests that contact with cows and straw and farm milk is protective for asthma, fodder storage and manure for atopic dermatitis, but neither of these explain the effect on allergic rhinitis. So it really depends on what your exposures are, what the protection you get. So the respiratory microbiome, we see a lot of the same thing. For example, the Copenhagen prospective study on asthma. The Copenhagen prospective study found that hypopharyngeal colonization at 1 month of age with either strep pneumoniae, H. flu, or M. catarrhalis in infants of mothers with asthma is associated with more frequent disease and more severe disease. And 1/3 of these subjects with colonization had asthma at age 5, compared to about 15 to 20% of patients without colonization. And poor control of asthma is associated with greater bacterial burden. And the bronchohyperresponsiveness is actually correlated with diversity. Microbiota, similar to other patients, is quite different in asthmatics. They tend to have more of the proteobacteria like H. flu compared to more Bacteroides in non-asthmatics. There's a particular type of bacteria that can be present, and I'm not going to try and pronounce that, in the respiratory microbiome that can break down steroids. And there's some question about whether that has a role in steroid-resistant asthma. Skin microbiome, some of the very same changes. Decreased diversity appears to be associated with increased disease. And interestingly, lesional skin shows temporal shifts in the bacterial composition associated with the flares and with treatment. So with flares, you get increased Staph aureus and Staph epidermidis with flares. But it's not clear if Staph epidermidis is there to fight off the Staph aureus or help them out. And Staph epidermidis may actually be an antagonist for the Staph aureus. It does induce anti-inflammatory cytokine release from keratinocytes, again through TLR2 binding, so the innate immune system is involved. And then you start to see more strep and beyond bacteria and corynebacteria after treatment. So you get kind of a restoration of normal response. Dietary associations with atopic disease. There are differing incidents of asthma in the UK and Mediterranean countries. Can't really be explained by antibiotics or birth method. So the assumption is that the diet may be playing a role. Increased fatty acid consumption is associated with atopy and asthma. Higher dietary antioxidant intake, lowered incidence of asthma, and the anthroposophic lifestyle, where patients sort of shun vaccinations and antibiotics and more fermented foods and vegetarian diets with high fiber, have less atopy. But that effect, that protective effect of their lifestyle, is negated by antibiotic use in childhood. So this is just a summary of some of the studies on the association between the gut microbiota and atopic disease and obesity in infants. So how do we treat it? What are we going to do about it? Well, breastfeeding protects against atopic disease, but may not be the case in allergic mothers. C-section delivery and antibiotic use prenatally or in infancy is associated with an increased risk for atopic disease, so you promote breastfeeding and vaginal birth. Prebiotics, probiotics, symbiotics. That's what everybody's doing now, and all these studies are being done. Cochrane Review suggested that there's no significant protective effect other than eczema with these, but there are actually some newer studies that are coming out that maybe show some effect. Some of the problem is that they're all done with different bacteria. They're all done for different durations. They either treat the mother or the infant only, but not necessarily both. So there's no consistent studies done with a defined bacterial colony. There are effects, though, on the immune system that you can see in murine studies. So you can actually measure these effects with probiotics. And immune effects have been noted in human studies, but haven't really translated into clinical effects. Most of the studies are flawed. They have limited microbiologic assessment, heterogeneity of study participants. You can understand from what we've talked about, if you don't control for method of birth and method of feeding, it's going to really throw off your effects. And also, some of the recent studies suggest that the manufacturing process and the food matrix used to deliver these probiotics may have an effect. There are studies that have shown some effect. It depends on family history. Mostly the effect is for eczema, perhaps allergic rhinitis. And there's really only one study that's shown a benefit for asthma and several that haven't. It appears that treating prenatally plus postnatally is important. Or prenatally alone, but just postnatal supplementation doesn't appear to have an effect. And cow's milk allergy, a recent study suggested that treating a hydrolyzed formula with the addition of lactose, most hydrolyzed formulas are lactose-free. And if you add lactose to the hydrolyzed formula, you get a more normal microbiota. It hasn't translated necessarily to clinical efficacy, but it does end up with better results in the microbiota. There's a significant decline in symptom scoring and increased quality of life in adults with atopic dermatitis. In this particular study, associated with increased Treg cells, increased Th1 response, decreased Th2 response. So there was a randomized, double-blind, placebo-controlled trial, 16 weeks of probiotics in adults with seasonal allergic rhinitis. No clinical difference in symptoms, but there were changes in the nasal mucosa on an immune basis between the control and placebo. Pacifier cleaning method, I was talking with Alan before that. There's a study out of Sweden where they looked at the incidence of atopic sensitization and allergies in a large study, and they looked at a small cohort of that study and analyzed pacifier cleaning technique and the incidence of allergic disease. And parents who clean the pacifier by sucking on it, their children had less allergic disease, significantly less allergic disease, which would fit with the microbiome sort of thing that we're talking about. And symbiotic studies, there aren't very many of those, but one did reduce the incidence of eczema at age 2, but not at older ages. Vitamin D, always got to talk about vitamin D when we talk about atopy. Microbiota actually affects the distribution and expression of the vitamin D receptor in the GI tract. Mice that lack the receptor have chronic low-grade GI inflammation and decreased T-cell homing. And vitamin D stimulates antimicrobial peptide production by intraepithelial cells, or intestinal epithelial cells. So where do we go from here? What's the problems? Well, we need to define the normal, healthy human microbiome, and that hasn't been done yet. We need to research the effects of individual microbial products. Is it the microbe itself, or is it a product that this microbe makes that makes a difference? Is it interaction between two microbes that results in a metabolic product that makes a difference? I mean, these are all questions that we don't need to know and need to find out. We need to research the effects of the mucosal microbiota versus luminal microbiota. Most of the sampling that's been done have been of luminal cells, and that may be very different from the mucosal bacterial cells. And we need to refine the mechanism to allow sequencing of large amounts of genetic material.
Whoa. Have they looked at endoscopy samples or bronchoscopy samples to see about the versus the epithelial?
Well, they haven't been able to, they haven't really done any endoscopy studies that I'm aware of. And most of the respiratory microbiome studies probably do involve bronchoscopy, but I don't think they're able to separate those out just by the techniques involved in every sample.
There's a lot of the microbiome we didn't talk about. Does anybody studying the viruses, parasites,
Mostly on it. Well, the fungi. There have been some studies on the fungi. Most of what you get out of there is bacterial. So there is fungi DNA, of course, and there's yeast DNA, and a lot of these studies were related to that as well. I just didn't have time to go into all of them. But there's a normal fungal, yeast colonization, and absence of that leads to disease.
Is there a normal viral?
None of the articles that I read really talked about it, other than in passing. They didn't talk about a normal viral. They talked about the pathogenic viruses. Didn't talk about normal virus.
They give up on the parasite stuff, like in the past. If you got trichurus or whipworm, you're going to get asthma, and if you didn't get that, you won't get asthma. Has that all been dropped in this microbiome stuff?
Yeah, there wasn't really a lot of talk about parasites because you're going to get so much environmental differences. It's difficult. I mean, in general, populations that live in areas of high parasites have lower allergic disease. So,
Were they giving the babies born of C-sections, the mother's vaginal fluid is like a little balm over their body? Is there any study where they looked at that?
Well, there may be people beginning studies like that, but it's interesting that the vaginal microbiome changes during pregnancy. And so you may lose some of those changes after delivery, and that's, of course, all related to hormonal effect. You get much less diversity in the vaginal microbiome during pregnancy with an increase in lactobacillus species. So they're trying to replace the species, but the problem is, again, that they haven't been able to develop techniques that adequately look at species level or strain level. And there may be a difference between different strains of the same species. So still something that they need to define.
Did you read much about the studies where they're trying to sterilize the gut and replace it with different bacteria to try and treat rheumatologic diseases and autoinflammatory
I specifically didn't. I mean, there's so much information on immunology and allergy. I didn't have time. There's a whole other list of autoimmune disease, obesity, and inflammatory bowel disease that related to this. Yeah.
Okay.
What about sequential studies, like somebody's got microbiomes wiped out by antibiotics, and then have they followed them sequentially? Do they repopulate, let's say, an adult, what they had before, or is it depending where they are geographically? What if they moved?
There are the adults actually recover fairly quickly, but it's of course diet and environmental dependent to some extent. Children recover much less quickly, and some of the changes in infants and young children may persist for up to a year afterward. The problem is you not only kill off a lot of bacteria, but you change the balance. And while you may get some of the bacteria back, it's not clear that you get the balance back right away and what effect that has. But again, that is dependent on